you open your refrigerator door, the light clicks on, and you probably feel this immediate sense of safety.
Oh, yeah, absolutely.
Right. Like the cold air hits your face, and you just instinctively trust that whatever is sitting in there is protected, it's preserved. I mean, we basically view the fridge as a microbiological pause button.
Yeah, because we are taught from childhood that cold stops bacteria. You keep the milk chilled so it doesn't spoil. You refrigerate the leftovers so they don't grow mold.
Exactly.
It is the golden rule of food preservation.
But what if that humming white box in your kitchen isn't a safe zone at all? Like, what if, to one specific, highly adaptable microscopic organism, your fridge is basically just a luxurious high-end incubator?
Yeah, that is the terrifying reality.
Today, we are diving deep into the research on Listeria monocytogenes.
Which is a pathogen that completely shatters our basic assumptions about food safety. I mean, it forces us to rethink everything from how we store deli meat to how we clean factory floors.
So our mission for this deep dive today is to unpack how this single, bizarre organism rewrote the rules of global food safety, totally transformed prenatal medical care, and actually taught scientists how human cells work.
It's a massive story.
It really is. And I want to be clear to you listening, this isn't your standard biology lesson. We are looking at a wild medical detective story. I mean, it involves slime molds, hijacked cell skeletons, and a decades-long identity crisis.
Well, Listeria is fundamentally a master of deception, and by understanding its biology, how it survives, how it moves, how it hides, you will see the underlying logic behind the strict dietary guidance given by doctors.
Right.
And those massive multi-million-dollar food recalls you always see on the news.
Yeah, I want to start with that identity crisis, actually, because the history here is just wild. For the longest time, nobody even considered this bacteria to be a threat to human food. It took decades just to figure out its name.
Right, yeah. Let's go back to the 1920s. So there was this dual discovery happening on two entirely different continents.
Oh.
In 1926, a team of researchers at Cambridge, Murray, Webb, and Swan, they were investigating a mysterious die-off in their laboratory rabbits and guinea pigs.
Just a total lab animal plague.
Exactly. And when they examined the blood of these sick animals, they noticed a massive, unexplained surge in a specific type of white blood cell.
A monocyte.
Yes, a monocyte. So they named the bacteria after that one bizarre symptom, bacterium monocytogenes, meaning monocyte producing.
Right, because they assumed it was just a strange rabbit bug.
Totally. Meanwhile, in 1927, a South African researcher named James Hill Harvey Peary was investigating a totally different die-off.
In gerbils, right?
Yeah, he was looking at liver necrosis in gerbils used for plague research, and he isolated an organism and proposed calling it Listerella hepatolytica to honor Joseph Lister, the pioneer of antiseptic surgery.
Which makes sense. But then eventually the scientific community realizes the Cambridge rabbit bug and the South African gerbil bug are the exact same organism.
Yep, they put it together.
But when they try to combine the research, Peary gets hit with a taxonomic roadblock. And I was reading this in the source material and actually laughed out loud. The name Listerella was already taken.
Yes, it was.
By a slime mold.
Literally, a slime mold beat him to the patent office, essentially.
That is so funny to me.
So, in 1940, Peary proposed a compromise. He modified his genus name to Listeria, and they attached the monocytogenes from the Cambridge team as a nod to that white blood cell reaction.
But even with a finalized name, Listeria spent the next 40 years viewed primarily as an occupational hazard for farmers and veterinarians. Like it wasn't a food thing at all.
No, not at all. It caused something called circling disease in livestock.
Oh, right, where a sheep or a cow gets a brainstem infection and literally walks in endless circles until it collapses.
Exactly. And humans only caught it if they were farmhands or abattoir workers who were constantly covered in animal fluids.
So it was a farmyard problem, not a kitchen problem.
Right. The connection to the human food supply, that required an entirely new level of epidemiological detective work.
So when did that shift happen?
The turning point finally happened in 1981 during an outbreak in the maritime provinces of Canada.
Okay, what happened there?
An epidemiologist named Walter Schleck was called in to investigate a cluster of human listeriosis cases. And tragically, this cluster included several stillbirths and neonatal deaths.
Man, that's awful. But how do you even trace an invisible bacteria from a sick patient back to a specific farm? It seems impossible.
It's incredibly hard. Schleck used a really rigorous case control study. He painstakingly interviewed the families of the victims about every single thing they had eaten in the months prior.
Wow.
And then he compared those diets to healthy people in the same region. And the statistical anomaly that kept popping up was coleslaw.
Coleslaw, of all things.
Yeah. And he didn't stop at the coleslaw, though. He traced the cabbage used in that coleslaw back to a specific regional farm. And when he investigated that farm's agricultural practices, he found they were fertilizing their cabbage crops with raw, composted manure from a flock of sheep that had recently been infected with listeria.
Oh, wow. So there is the circling disease connection right there.
Exactly. That was the smoking gun. It proved that listeria could survive in the soil, transfer to animal manure, contaminate a vegetable growing in the ground, survive the harvest, and make it all the way to a human dinner table.
It completely rewrote the definition of the bacteria overnight. Suddenly, it's a foodborne pathogen.
Yes.
But, the reason it makes that jump from the dirt to your dinner plate so successfully comes down to this biological superpower. It breaks that golden rule of the refrigerator we talked about earlier.
Right. The scientific term for this is psychotrophic, meaning it thrives at refrigeration temperatures.
Which is wild because most bacteria go dormant in the cold.
They do. To understand why, think of a stick of butter. At room temperature, it's soft. In the fridge, it turns into a solid brick. A normal bacterium cell membrane does the exact same thing. It stiffens up in the cold, locking up the cell's machinery so it can't eat or divide.
But Listeria doesn't freeze up.
Not at all. As the temperature drops, Listeria alters its own chemical structure.
Really?
Yeah, it pumps out specific short-chain fatty acids that keep its cell membrane fluid and flexible. It essentially turns its membrane from butter into olive oil.
Wow, that is incredibly clever.
It is. Because of this, it can actively grow in environments as cold as -1.5 degrees Celsius.
Which is below the normal freezing point of water?
Right. Usually it survives that in high-salt foods.
Yeah.
And it can survive all the way up to 45 degrees Celsius.
That is just a massive range.
Yeah.
I mean, think about the implications of that for your kitchen. If you buy a package of deli turkey that happens to have a few Listeria cells on it, and you leave it in your perfectly functioning fridge for 3 weeks,
Yeah.
that meat isn't just maintaining its bacteria level. It is actively cultivating more of it. You are literally farming a pathogen in your crisper drawer.
It's unsettling. And it completely upends the advice we normally give. We usually assume extended cold storage equals safety. For Listeria, extended cold storage increases the danger.
So that is why public health officials tell high-risk groups not to trust the fridge when it comes to things like cold cuts.
Exactly. The rule becomes, when in doubt, throw it out.
This cold-loving nature also creates a nightmare for tracking outbreaks, doesn't it? Because it stretches out the timeline.
Oh, absolutely.
Like when someone gets normal food poisoning, say, from salmonella on undercooked chicken, they are intimately acquainted with their bathroom within 12 to 24 hours.
Right, you usually know exactly what meal made you sick.
But Listeria plays a much longer game.
A much longer game. Its incubation period can range from a single day to an astonishing 70 days.
70 days.
Yeah, though it usually hits between 1 and 3 weeks.
70 days. I mean, I can barely remember what I had for lunch on Tuesday, let alone a specific sandwich I ate 2.5 months ago.
Put yourself in the shoes of an epidemiologist trying to solve that puzzle.
It sounds impossible.
You have 50 patients scattered across 3 states. You have to synthesize 2 months of their grocery receipts, restaurant visits, and snacking habits, hoping to find a single common denominator.
That slow incubation is really its greatest weapon for evading public health detection.
It is.
But, okay, it evades detection outside the body. What it does once you actually swallow it is pure sci-fi horror.
It really is.
This is where it goes from being a public health nuisance to a master biological hijacker.
Yeah. So to understand the invasion, we classify Listeria as a facultative intracellular pathogen.
Which means what exactly?
It means its primary survival strategy is to hide inside your own cells. Specifically, cells that aren't even designed to engulf bacteria, like the epithelial cells lining your intestines.
I want to slow down here because the mechanics of how it breaks into an intestinal cell are fascinating. It doesn't just smash through the wall.
No.
It picks the lock.
Precisely. It uses 2 specialized surface proteins called internalin A and internalin B. Let's focus on internalin A. It acts like a counterfeit key that perfectly fits into a human receptor protein called E-cadherin.
The source notes describe E. cadherin as the molecular Velcro that holds your intestinal cells tightly together. So it forms a solid wall so nothing leaks out of your gut.
That's a really great way to picture it. Listeria basically mimics the other side of that Velcro.
Oh, wow.
So when it bumps into your intestinal wall, the human cell thinks it's just connecting to a neighboring cell.
Like it's tricked.
Totally tricked. It embraces the bacteria, zippering its membrane around the Listeria and pulling it inside a protective bubble called a vacuole.
Wait, getting trapped inside a vacuole sounds like a bad move for the bacteria. Isn't that how a cell normally digests and destroys invaders?
If it stayed there, yes. The cell would flood that vacuole with acid. But Listeria drops a chemical bomb first.
Of course it does.
It secretes a toxin called Listeriosis O, or LLO. This toxin acts like a microscopic hole punch. It shreds the membrane of the vacuole, allowing the bacteria to slip out into the main body of the cell, the cytoplasm, before the acid can destroy it.
So now the bacteria is sitting in this warm, nutrient-rich cellular soup, completely hidden from the immune system outside. But it doesn't just sit there. It starts building a propulsion system.
This is, hands down, the most famous part of its biology, the actin rocket.
The actin rocket. Okay, explain this.
Once it's free in the cytoplasm, the bacterium produces another surface protein called acta.
Okay.
And this protein's entire job is to hijack a piece of your cell's machinery called the ARP23 complex.
Which does what normally?
Normally, your cells use ARP23 to build their own structural scaffolding out of a protein called actin. Listeria tricks this machinery into rapidly building a rigid, directional comet tail of actin filaments right behind the bacteria.
It literally builds a rocket engine out of human scaffolding.
It does.
I keep picturing like a burglar who picks the lock to your front door, hangs out in your living room, and then takes your own home renovation supplies, your wood, your nails, your power tools.
Yeah.
And builds a high-speed tunnel straight through the drywall into your neighbor's house.
The burglar analogy is spot on. That actin rocket propels the bacteria forward at several micrometers per minute.
Which is fast for a bacteria.
Very fast. It rams with such force that it pushes the membrane of its current host cell straight into the neighboring cell.
Oh, yeah.
And then the neighbor cell engulfs that protrusion, and the Listeria is immediately inside a new host, ready to start the cycle over again.
Bypassing the police patrolling the streets outside entirely.
Exactly. It never enters the bloodstream during this transfer. It remains completely invisible to your circulating antibodies.
That is terrifying.
It is. But in a fascinating twist of scientific fate, scientists in the late 80s and 90s, like Daniel Portnoy and Julie Theriot, they realized they could use this burglar mechanism to study human biology.
Oh, really?
Yeah. Listeria became the foundational model for understanding how the ARP23 complex works.
Wow.
By watching this pathogen hijack our cells, scientists actually figured out the fundamental mechanics of how all animal cells move, change shape, and divide.
Well, that's an incredible silver lining.
It really is.
But that stealth mechanism, the ability to hide inside cells and travel without touching the bloodstream, that is exactly what makes it so devastating to one specific group of people.
Pregnant women.
Right.
The statistics are stark. Pregnant women are roughly 10 times more likely to contract listeriosis than the general population.
And there is a major misconception here we need to clear up. Because when you look at food safety warnings, pregnant women are usually lumped in with the elderly and the immunocompromised.
Yeah, they are.
And people assume it's just because pregnancy makes you generally fragile or tired.
Which is a fundamental misunderstanding of the biology. I mean, pregnancy is not an illness.
Right.
But to successfully carry a pregnancy, the mother's body has to naturally downregulate certain cellular immune responses.
Okay, why is that?
Think about it. A fetus is genetically half foreign tissue.
Oh, from the father.
Exactly. If the immune system operated at full aggressive capacity, it would recognize the fetus as an invader and attack it.
That makes total sense.
Hmm.
So the body intentionally lowers its guard in specific ways to create this beautiful, localized immune tolerance for the baby.
Unfortunately, that necessary tolerance creates an opening. Because the specific cellular defenses that usually hunt down intracellular hiders are dialed back, Listeria can multiply completely unchecked.
And it doesn't just stay in the mother's gut.
No, it can cross the placental barrier.
That's the scariest part.
Obstetricians actually monitor a specific group of pathogens known by the acronym TORCH.
Torch. What does that stand for?
It stands for toxoplasmosis, other agents, which includes listeria, rubella, cytomegalovirus, and herpes. These are the rare infections capable of crossing into the fetal environment.
And when Listeria does this, the results are catastrophic.
They are. It causes a condition called granulomatosis infantiseptica.
Wow, that's a mouthful.
It is, and it's characterized by widespread fetal abscesses. It frequently leads to miscarriage, stillbirth, or severe neonatal sepsis.
I mean, almost an entire section of the standard prenatal diet pamphlet exists because of this one organism.
Pretty much.
And the specific foods they tell you to avoid, soft cheeses, deli meats, those weren't chosen at random. They were dictated by massive, tragic outbreaks.
Yeah. The turning point for dairy was the 1985 Jalisco cheese outbreak in Southern California.
What happened there?
A factory producing unpasteurized milk-style Mexican soft cheese was contaminated. The outbreak caused at least 142 cases and 48 deaths.
God, that's awful.
And the overwhelming majority of those deaths were fetuses and newborns.
That is heartbreaking.
It was, and that single event cemented the modern rule. Avoid unpasteurized dairy and soft cheeses during pregnancy.
But the threat isn't just animal products, right? Because the 2011 Jensen Farms cantaloupe outbreak completely shattered the idea that you only need to worry about raw meat and cheese.
It really did. That outbreak remains the deadliest foodborne illness event in modern U.S. history.
Really? From cantaloupes?
Yes, killing 33 people. The idea that a whole fresh fruit could harbor a deadly pathogen caught a lot of people off guard.
I mean, how does a whole melon become a lethal vector? You don't exactly think of bacterial meningitis when you slice up a fruit salad.
It comes down to the physical properties of the cantaloupe itself. Look at the rind.
Okay.
It's heavily netted, porous, and deeply textured. It is practically impossible to fully sanitize.
Oh, so it just hides in those little grooves.
Exactly. Furthermore, the melon's flesh isn't highly acidic, so if bacteria get inside, they face no chemical resistance.
Right.
At Jensen Farms, a combination of dirty processing equipment and pooled unchlorinated wash water allowed Listeria to contaminate the rinds of thousands of melons, which then shipped across 28 states.
Wow. Which perfectly transitions into the industrial side of this. Like, if Listeria can survive on a melon rind or a deli meat slicer and it grows in the cold, how on earth do you regulate it in a massive, modern food processing plant?
It's incredibly difficult. You are fighting an enemy that actively builds fortresses in the environment.
Fortresses?
Yeah. In a food plant, Listeria doesn't just float around as single vulnerable cells, it forms biofilms.
What exactly is a biofilm?
Imagine a microscopic city encased in a protective matrix of slime.
Gross.
The bacteria anchor themselves to a surface, often stainless steel conveyor belts or the inside of cold, damp floor drains.
Right.
And they secrete this resilient barrier. Once a biofilm is established, routine cleaning and standard sanitizers often just can't penetrate it.
That is insane.
The colony can persist in a factory for years, periodically breaking off and shedding bacteria into batches of passing food.
And this was the major factor in the South African polony outbreak in 2017 and 2018, wasn't it?
Yes, the largest listeriosis outbreak ever recorded globally. Over 1,000 confirmed cases and more than 200 deaths.
Over 200 deaths?
Yeah. It was traced back to a processed meat product called polony, manufactured by Enterprise Foods. And the investigation proved that Listeria's true favorite habitat isn't necessarily the food itself.
It's the factory.
It's the cold, damp machinery of the factory. The factory equipment is the actual vector.
Okay, knowing all of this, that it hides in invincible slime fortresses, multiplies in the fridge, and takes 70 days to show up in patients, how do governments legally handle this? Because I know this has sparked a massive ongoing debate in global food policy.
It has, and it really centers on two completely rival regulatory philosophies.
Okay, what are they?
In the United States, the FDA and USDA enforce a strict zero-tolerance policy for any ready-to-eat foods.
Right.
If an inspector detects even a single cell of Listeria monocytogenes in a product that won't be cooked before consumption, it triggers an immediate massive recall.
Which, I mean, makes sense to me. If it grows in the fridge, any amount, even one cell, is a ticking time bomb. Why would anyone do it differently?
Because zero tolerance is practically impossible to maintain without massive collateral damage.
What do you mean?
Well, the European Union operates on a risk-based threshold.
Okay,
They acknowledge that Listeria is everywhere. It's in the soil, on farm boots, in the drains. You can't escape it.
fair point.
So they legally permit up to 100 colony-forming units per gram in certain ready-to-eat foods,
Wait,
provided the food has a short shelf life or properties that make dangerous multiplication unlikely before it spoils naturally.
letting any amount of a pathogen that causes fetal abscesses into the food supply sounds incredibly reckless.
Critics of the EU model completely agree with you. They argue you are playing roulette with vulnerable populations.
Right.
But the proponents of the EU model argue that the American zero-tolerance policy creates staggering, unnecessary economic waste.
Oh, I see.
When the U.S. detects a microscopic trace amount of Listeria that might never grow to a dangerous level, they force companies to throw out thousands of pounds of perfectly good food.
It's a brutal dilemma, isn't it? Like you have pure economic pragmatism and food waste on one side, and the terrifying, unpredictable biology of a cold-loving pathogen on the other.
And because the bacteria is so adaptable, the debate remains totally unresolved. You cannot sterilize the natural world, but you also cannot predict exactly how Listeria will behave in every single refrigerator.
Let's zoom out and look at the sheer scale of the journey we just took.
It's a big one.
We started with an obscure 1926 rabbit infection, an organism that couldn't even figure out its own name because a slime mold beat it to the punch.
Yeah, from a farmyard veterinary curiosity to a world-class foodborne pathogen. I mean, it defied the fundamental laws of refrigeration by turning its cell walls into liquid gold.
Olive oil, basically.
Right.
It hijacked our intestinal Velcro, punched holes in our cellular traps, and built rocket engines out of our own cell skeletons.
It manipulated our biology so flawlessly that it accidentally gave scientists the blueprint for how human cells move.
And it forced governments to rewrite international agricultural policy.
The scale of its impact, from the molecular level to the global supply chain is just truly staggering.
It is. And there is one final lingering thought from the research that I want to leave you with.
Okay.
We talked about that cell-to-cell actin rocket mechanism, the burglar tunneling through the walls.
Yeah.
That exact mechanism is now being used in laboratories around the world as a model system to study cancer cell migration and even neuroscience.
Oh, wow.
Right. The implications there are profound. How cancer metastasizes and spreads through the body relies on very similar actin dynamic.
That is fascinating.
So think about it. The exact same molecular trick that makes this pathogen such a terrifying stealth killer, the reason pregnant women have to avoid soft cheeses.
Yeah.
That trick might ultimately be the key that helps scientists unlock how cancer spreads or how the neurons in our brain physically function.
Could the ultimate foodborne villain accidentally hand us the blueprints to cure completely unrelated diseases?
It is the ultimate paradox of microbiology.
Yeah.
Sometimes the organisms that harm us the most are the ones that end up teaching us the most about ourselves.
That is a wild thought to end on.
It is. So the next time you open your refrigerator door and that light clicks on and you feel that comforting chill, just remember,
Yeah.
the cold isn't always as quiet as it seems.